here is time. And you can see between hours 2 and 3, I was able to drop the amount of oxygen on the green side, and then reverse the gradient on the left side by dropping oxygen in the purple arena. And so we would observe the fish as it went from arena to arena, and just sort of just take notes of how long it inhabited each side. And so here's what an ideal sort of situation would look like if we have time on the right side, so time in this green arena. If the oxygen concentrations are similar or identical, we expect about 50% inhabitants on the right side arena, right? So it shouldn't care. You go left and right, and because oxygen is the same and there's no avoidance, if there's no oxygen difference, 50%. If we make the right side hypoxic, we expect this animal to inhabit the purple arena or spend less time on the right side. And if we make the left side, and if we make the left side hypoxic, on the purple side, we expect more time to be spent on the green side, so we expect a larger bar on this side. So this is what our ideal data would look like. And if we calculate the avoidance for each side, we took an average there. Say it's 80% avoidance when it's on the right, 90% avoidance when hypoxia is on the left. The average would be about 85%. And so this number indicates the degree of avoidance, where 100% would mean this animal completely avoids hypoxia. 50% would mean it doesn't care. It's spending equal time on both sides. And 0% would mean it's seeking hypoxia, so it's going to these hypoxic regions. And so we did this with and without that that net to allow or deny air breathing. And so here's a schematic of what happens. So here's our theoretical tank again, with the dark blue indicating hypoxia, and this would be normal, I guess, like high oxygen water. And this is a fish with the ability to breathe air, and this is the fish without the ability to breathe air, so it's underneath this net. And if we look at 75 millimeters of mercury, this is our first test, we looked at avoidance. You can see there's almost no change between left and the right side. So there's an avoidance score of 51%. So this fish would inhabit the high oxygen zone, and would also inhabit the low oxygen zone. There's really no preference when the gradient is at 75. For an animal without the ability to breathe air, we see almost identical results. So no avoidance, about 50%, so exactly identical. And here it is here. So inhabiting both sides, or hypoxic and normal water. Hypoxic and high oxygen water. So maybe it's not hypoxic enough, so let's drive it down a little bit more. So we're going to test at 15 millimeters of mercury. And we look at the results again for a fish without the ability to breathe air. And we do see some change in behavior. However, this turned out to be non-significant. So about 59% avoidance behavior. So it will inhabit on the more hypoxic side of this level of oxygen right now. And if it doesn't avoid it when it can't breathe air, I think it's safe to assume that it won't avoid it either with the ability to breathe air. So we didn't test that. But we instead decided to try one more oxygen level, so about 37 millimeters of mercury. And once again, we don't see any change in behavior here. So no avoidance, about 54%. And so maybe we should drive it even lower, but I really couldn't, because I would blow through a tank of nitrogen in a day. So we just said there's no hypoxia avoidance for what we see for these two animals. And just to make sure that this level of hypoxia was probably a good level to test, I went through the literature and I looked at what is hypoxic for this animal. And we can look at air breathing threshold, and it's about 60 millimeters of mercury, so this animal does want to breathe air. Even if it can't, it still doesn't seem to avoid hypoxia. The P-crit, so this is the level of hypoxia in which the animal starts decreasing its activities because it can no longer consume enough oxygen for it to, to maintain function. That's about 55 millimeters of mercury, that's here. And then 60% mortality after 24 hours of no air access is 40 millimeters of mercury. So I think this level of 37 millimeters of mercury in terms of testing for hypoxia avoidance would be enough to elicit a behavior if there was a behavior present. And so just to test out that my setup wasn't, I guess just wouldn't show behavior even if there wasn't present, I used another fish. And so this is our sort of canary in the coal mine, because I know this fish has a strong reaction to hypoxia, and it's called Polypterus. And so I did the same test, and with the ability to breathe air, no avoidance. So he's going to inhabit this side. But once you deny his ability to breathe air with this net, you do see a strong avoidance pattern here. And so this is statistically significant. And we have avoidance behavior of 86%. And so a fish over here is not like it. And it's going to behave like a water-breathing fish and hang out in the high oxygenated water. So I guess this study here, using this other fish, really did validate that if this fish, Pangasius, had avoidance behavior, we would have seen it. And why it doesn't, or it has no hypoxic avoidance behavior, and Eric says has no effect, and why it doesn't, I think, is because it has this natural ability to inhabit the surface in nature. So if it can always access the air, why would it avoid hypoxia? So it's sort of like, I guess, you going out and worrying about, a scuba diver worrying about how hypoxic the ocean is, right? It doesn't really matter because you can take your own air with you. So this fish probably has lost the ability to avoid hypoxic air simply because it wouldn't really have this net in its natural environments, and it never really is denied the ability to breathe air. So if we look at hypercapnia, and we have the same schematic here where we have hypercapnia in this deep red section here, and normal water or low CO2 water in this lighter section here. And we look at 38 millimeters of mercury. So we don't see any avoidance without the ability to breathe air, and so about 54%. And so this fish is likely, excuse me, it's likely to inhabit or will inhabit waters below this level. And it's safe to assume, I think, without or with the ability to breathe air, that they would change the behavior. And so I didn't test without the ability to breathe air, or with the ability to breathe air for this fish here. But I did at 57 millimeters of mercury. And so with the ability to breathe air, we actually didn't see any avoidance. We see some change in behavior, but it wasn't statistically different. So avoidance was about 61%, again, non-statistically significant. And then without the ability to breathe air, he'll inhabit the lower portion or the more hypoxic region. Without the ability to breathe air, we do see some change in behavior, but again, it wasn't statistically significant. So this fish, again, doesn't show any avoidance at 57 millimeters of mercury. Or it shows some, but it wasn't statistically significant. So it's not a black or white response. Some fish can, some fish will, some fish won't. We expect sort of a gradient. But in terms of statistical significance, we don't really see it showing up. So I pushed the CO2 all the way up to 130. And at this level, we see almost full avoidance here. So finally, we have hypoxia avoidance, almost 90%. So this fish strongly avoids hypoxic regions, even with the ability to breathe air. And so I think it's safe to assume without the ability to breathe air that hypoxic avoidance wouldn't be the threshold wouldn't be any higher. So we'll put this fish on this side of the line as well. And so if we look at what these levels are in nature, or in aquaculture ponds, here we have what's harmful to the trout. Here we have an aquaculture pond at 38%. And here we have their upper natural range, or just any range in nature, rarely exceeds 60 millimeters of mercury. So what we see is not really a hypoxic avoidance or hypercapnic avoidance behavior at any really ecological relevant level here. So it does have it, but not any level you would expect to see in nature. But the level we're really interested in is this aquaculture pond level, so 38 millimeters of mercury. And we don't see avoidance at 38. We see slight avoidance, again, around 60%, not significant, at 57. But in terms of aquaculture and the relevant level here, we don't see hypoxia or hypercapnic avoidance. So CO2 levels in aquaculture are not avoided, and this level likely represents a benign concentration for Pangasius. So for behavior, at 38 millimeters of mercury, we really don't see any avoidance behavior. So lastly, we're going to look at stress biomarkers to see if these animals are expressing any type of stress that we can be measured by taking blood samples. And so just to describe what stress is, here we have an animal in homeostasis, so it's a stable body condition. It's a happy fish. Then, environmental stressor comes and disturbs the animal. We have stress. And an example of that is hypercapnia. So if this fish is in hypercapnia, say homeostasis, in this instance is going to be pH. As we saw earlier, high CO2 in the blood lowers pH in the blood, so we have a respiratory acidosis. That's going to be the stress. And this animal is going to elicit a stress response. And what that is a fight or flight response. And in this instant, for hypocapnia, it's going to be flight. And these are just typical stress indicators. We have glucose, lactate, and hematocrit, which we all expect to increase in stress. And what this is poisoning the animal for metabolic activity, or in this case, flight. So it wants to move out of the area. And so glucose in the blood will increase in the blood. That way it's got metabolic fuel ready to shove to the muscles so it can swim out of these areas. Lactate is an indicator of that muscle activity has already occurred, so that there's some agitation and some activity, maybe an escape response. So lactate is the indicator of stress, and that activity has already occurred. Hematocrit is the concentration of red blood cells in the blood. So in stressful situations, animals increase hematocrit. That increases the oxygen carrying capacity of the blood, and so they can deliver oxygen more readily to active muscles in a fight or flight response. So we expect compensation after a stress response. So for this animal, moving away from the area would be its compensation for hypocapnia, and it would remove the environmental stressor, and homeostasis would be reestablished. However, in situations where this animal can escape, such as in an aquaculture pond where the whole thing is hypocapnic, stress can become maladaptive. So this is an animal stress response as it swims around, it spends a bunch of energy, and in that case, instead of recovering from stress, it creates stress on its own. So now this animal has just expended a bunch of energy, and it becomes maladaptive because it's not knocking off this environmental stressor, there's no compensation going on. Instead, you have a loss of energy input into reproduction and growth. And so animals in stressful conditions reallocate resources from long-term investments in growth and reproduction and push them towards more immediate needs, such as compensating for environmental stressors. And so as a farmer, this is what you want to avoid. You want to reduce stress in your system so that you can invest more in reproduction and growth for your animals, or your animals can invest in growth and reproduction. So I wanted to look at what happens when we introduce these animals to high CO2 environments. And so here's our experimental tank with a low CO2, and then we expose them to hypocapnia. We bubble in a bunch of CO2, raise the CO2 level in aquatic environments, and look at how stress indicators might respond. So if we look at, for example, glucose, we might expect low level of glucose before exposure, and then a high level of glucose after hypocapnic exposure if there is a stress response. Or if this just doesn't care, we expect no change. And of course, we ran a control where we didn't expose these animals to any high CO2 and compared that with the experimental condition. We took blood samples at 0, so before exposure, 3, 24, 48, and then at 72 hours. And so to do this, you can't really let a fish go, catch it, take a blood sample, let it go again, 3 hours later, catch it, because that's stressful for this animal. You'll see stress response just by catching, and releasing this animal and stabbing it with a needle. So what we have to do here is develop methods in which we could discreetly sample blood from this animal without disturbing it and without it noticing. So pretty much trying to remove stress from the situation, except for this hypocapnic exposure. And so fish have what's called the dorsa aorta, and this is sort of the central line of the fish, or the main blood vessel. And we're able to catheterize it, or put a small tube in there and able to pull blood through it. It's like a very long, flexible straw. And so here's a picture of me going through the roof of the mouth of the fish, right here with a guide wire. This is actually a guitar string that I filed down, and I could use to guide the catheter through the top of the mouth and into the dorsal aorta, like this. And so we fastened it to the top of the animal, so it comes out the nose, and we sutured it along the dorsal side of the animal, so it's secure. And then attached a float. And so the float was able to keep the catheter away from the fish, and then we could just come in and hook the catheter without having to chase the fish around. And so here's a video of the fish swimming around. You can see the little floats here. As the fish is recovering, and the floats are just trying to stay out of the fish. So this was actually a really hard experiment because it's not like a patient in the in the ER where we put a catheter in, they sit there. These fish swim around and try to get it tangled on stuff or get it sucked into the pump. So it really took a lot of effort on almost, I think, 30 surgeries to get enough for this study, to get big enough sample size for this study. And so here's what our ideal data would look like, and here's what our data does look like. So here's blood PCO2. You can see control in these white dots here is almost perfect, right? It looks just like the control here. In hypercapnic exposure, we see low blood PCO2 before exposure, and then after exposure, we see a significant elevation. So that just proves that we're able to make the blood and the water hypercapnic for the sample. If we look at glucose, we can see that there is no difference in glucose between control and experimental conditions, and experimental conditions after hypercapnic exposure and before hypercapnic exposure. So this really looks like no response. Looks just like this light blue trend here. If we look at lactate, again, no response. And if we look at hematocrit, again, we don't see any response. We don't see a difference before exposure and after exposure, or between control and non-control, or control and experimental. And so maybe this fish just doesn't respond using these parameters to stress. And so I just want to verify that these parameters are able to, you are able to stress the animal and see a response in these parameters. So we chase these fish around in the tank just to do some exercise-induced activity. And so here we see blood glucose at resting, and we see a significant increase after exercise. Here we see blood lactate, and here we see hematocrit. So this just verifies that these parameters are not only responsive, but the experimental levels to a high CO2 fish are in fact the same as what we would see for a resting or unstressed fish. So this fish, I think we're going to say it's not stressed, or at least all the indicators we measured, we don't see any stress. So this level of hypercapnia is not stressful for plantations. And so we're going to fill this in here and say stress biomarkers, no change. And so if we look at these all together and look at them in relation to each other, no oxygen uptake means there's no avoidance, no increase in blood lactate or glucose, means there's also no activity as well. So it's nice when everything agrees with one another. And the impact on health and production of this high CO2 environment is likely to have no effect here. And so I gave this practice talk yesterday, and somebody just shook their heads and, oh, that's really sad that your thesis is full of negative results. But I think in this case that negative results are the most interesting results for the mere fact that what this animal encounters naturally and on a regular basis is lethal to what we know about other fish here. So I think this negative result just shows that this animal has amazing capacity to cope with and adapt to a high CO2 environment. So not that sad. So in summary, we don't see any significant, this level of CO2 does not represent a significant physiological challenge for this animal. This animal is very tolerant of hypercapnia, and it seems to thrive in this environment. And that regulating CO2 below this level of CO2 is unlikely to have additional benefits for this animal in terms of industry use. So future directions. Of course, long-term studies needed to look at growth rates and food conversion ratios. But implications from this work, this work pretty much provides new physiological data on the effects of CO2 and different thresholds for this animal, and it's hopefully can serve as a foundation to be able to characterize really what the optimal growth conditions are for this animal. And it's really an example of where basic biology can hopefully have a real-world application in terms of being able to describe what conditions these animals should live in and how different changing operational aspects of industry can have effects on these animals. And so hopefully this can create sort of data-driven set of methods and policies by which the aquaculture industry can improve its efficiency. And just to come full circle, if we look at the state of the World Aquaculture Fisheries Report again, you can see aquaculture is important in the past, and it's going to be even more important in the future. And a lot of, excuse me. So hopefully this work will be able to increase the efficiency of this industry and be able to meet future food demands. And I think the World Report really states it best in that learning more about the biology of these animals can hopefully help meet our sustainable development goals for the future. And I just acknowledgements time, so thanks to my committee. This is Donovan, Jim, and this is Tim, and they've sat through this talk for the last 6 years, and so hopefully you haven't got bored of it. I just want to thank them for the input and guidance they've been giving me for the past 6 years, and especially thanks to Jim, who's given me a lot of latitude in terms of what I can do with my PhD and what I want to do for my work. He's pretty much bankrolled my entire thesis, and also given me the connections that I need to. So for example, he hooked me up with the team in Vietnam, and also different funding sources, and if I needed to learn a surgical technique, he told me he'd send me to Sweden, and so I've been to probably about 5 or 6 different countries, thanks to Jim, and for free. Thanks to other members of the Hicks lab. Last one standing, I guess. Well, Jim's still here. Maybe just the slowest one. But we had a lot of fun looking at different animals, so it's great to interact with these people who don't only work on fish, and be able to see sort of what physiology and gas exchange and research looks like outside of just, I guess my past was just looking at marine. I was at Marine Institute, so just looking at fish. So there's a whole world of physiology out there besides just fish. The teams in Denmark and Vietnam who really made my time in other countries at home. So this is Gam and Tung, who I worked with both in Vietnam, and also I met them both in Denmark too, as we both did about, I think, 3 months in Denmark. This is Mark. So Mark is sort of my other advisor away from home. So he pretty much set up the whole program for research in Vietnam, and was there and encouraged me to come, and also encouraged me to come to Denmark as well and work under him there. So it's been a great influence on my work and a big help as well. Thanks to the undergrad, so they've been really essential for me to do the surgeries and long-term studies, where you have to come sample in the middle of the night. I can just send one of them. They like going at night. So I tell myself. But in terms of doing surgeries too, I really needed a 4th and 5th hand, so they've been instrumental in just being able to hand me tools or being able to revive an animal while I can get the next one on the surgery table. So thanks a lot to them. And family and friends too who have supported me along the way. Thank you all. Right. And also like funding sources, so APS paid for me to go to Vietnam, company biologists paid for me to go to Denmark. And of course, UCI has paid for me to go to different conferences and also support me in terms of teaching fellowships and dissertation fellowships over the past 6 years. Thank you.